Hello and welcome to Dr. Varsha Gaitonde's channel. In today's video, we shall discuss about the 3K Rice Genome Project, popularly called as 3000 Rice Genome Project. Before understanding the rice genome and 3000 project, you should understand the basics of rice. As you know everything about rice, but some of the basic things I am going to discuss here. Rice is known for its tremendous within species and within genus genetic diversity because, as you know, it is the second most important consumable cereal crop across the world. So it is having around 230,000 germplasm accessions maintained in gene banks worldwide, mostly in of Asian origin. Chinese Academy of Agriculture Sciences, Beijing Genomic Institute, Shenzhen and International Rice Research Institutes are launching this particular 3000 genome program in order to get the tremendous improvement in genetic diversity as well as for improving specific kind of quality of rice. The 3000 Rice Genome Project is the major step towards revealing the genetic diversity in all of the world's rice germplasm collections. That means it is for revealing the genetic diversity. The another purpose of this is to estimate or extract the genotype to phenotype relationship in a better way. Why rice? Because as you know, it is the second most important crop. We have to improve at least 25% of its production and productivity by 2030 to feed the predicted population growth. This has to be achieved using less land because land can't be extendable, less available consumable water, under more severe environmental stresses expected due to the effect of climate change and disease pressures. The Giga dataset of publicly available genome sequence having about 14x depth of coverage derived from 3000 accessions of rice with global representation of genetic and functional diversity. The aim is to comprehensively and systematically mine the dataset to link the genotypic variation to functional variation with ultimate goal to create new and sustained rice varieties. So the panel of sequenced rice accessions represent diverse set of originating from 89 countries which have collection of about 13,000 Oriza sativa accessions, nearly 1,80,000 of rice accessions of some other species, and these are conserved in international rice gene bank collections, that is IRGC at IRI, and China National Crop Gene Bank, that is CNCG. This is the entire representation from where the rice accessions and are collected and how much percentage the regions are contributing. For example, Southeastern Asia contributed about 9 accessions. Likewise, from America, 22 accessions or 22% of the accessions are collected. Oceania, 4% accessions are collected, like that. Now coming to the practical applications of the outcome of 3000 rice genome project. The main application is to decipher global and local population differentiation, constructing new high quality reference genome representing major varietal groups, creating haplotype maps by LD and recombinant breakpoint analysis, then to build one or more pangenome assembles for each of the varietal groups and create annotation mappings between pangenomes. Here I am not going to explain you the individual terms like SNPs or pangenomes. These all are explained in our previous discussions. The last outcome or expected outcome is to discover SNPs, structural variants and indels between and within populations. So these were developed under the multiple institutions like with the help of Global Rice Science Partnership of the Consultative Group on International agriculture research for extensive and systemic characterization of phenotypes of accessions for a wide range of traits to discover important sequences and regions using genome wide association studies. Phenotyping coordinated by IRRI and CAS is in progress for biotic and abiotic stress tolerance, grain quality characters, plant development and yield traits, high throughput phenomics using image and sensor capture from controlled environmental and field based platforms. This will contribute immensely to the ability to associate sequence information with phenotype. These combined efforts will provide greater depth and coverage compared to other studies, yielding deeper insights and broader applications. This is about the five groups of the different accessions made, classification made by 3000 rice accessions. Five sets are made of 2 lakh random sets from 18.9 million discovered SNP variants. So the five groups are indica type, os and boro type, then basmati type, japonica type. Some are intermediate type, which are collected from temperate, tropical, intermediate characters. Now, the outcomes of this 3K rice genome project: new population specific genotyping arrays are developed, then detailed studies on population structure that have been shaped by evolutionary domestication and selection process. Third, by including lines used in mapping and breeding programs, we can target gene validation for direct use for trait improvement in breeding population. These breeding populations developed from the sequenced lines which enable implementation, testing and improvement of novel breeding strategies like genomic selection and recurrent selection in rice breeding programs. So, to give an example, I am quoting one success story, that is to develop rice accessions or to develop a rice variety with low glycemic index. As you know, glycemic index is directly related to diabetes, and there is a notion that by consuming high quantity of rice, people are becoming more prone to the diabetes condition. So, rice varies from lowest 52 to highest as 100 index of glycemic index. So, the main aim is or target is to develop the varieties which are having low glycemic index. Okay, so as I have already quoted, there are many, more than lakhs of accessions which are having variant kind of glycemic index. Here they are going to cross, or they are going to select the accessions which are having low glycemic index so that they can address the issue of diabetes, or they can recommend a variety which is even consumed in the diabetic conditions, or diabetic patients can consume it. So this is by the efforts of IRRI and other academies worldwide. Even Bill and Melinda Gates Foundation is also involved along with Chinese Ministry of Science and Technology in to get success in this project. So this is all about 3000 genome project in rice. So friends, if you have any doubt, just put it under the comment section. If you have any suggestions, that those are also welcome. Thank you for watching. If you like, please do not forget to subscribe. Keep on commenting, sharing. Thank you.